This is the third part in our fall outbreak webinar series, so feel free to watch the recordings of the first and second, and those will be up on our YouTube. And if you've made it to all three, thanks for joining us for each one. If this is your first one, welcome. So today we're going to be talking about what the BV-BRC is, the H5N1 outbreak, and the monkeypox virus outbreak, and some BV-BRC tools that you can use to analyze that data, and then any conclusions and questions. So as I mentioned, this is the third webinar in our three-part series. So the first one, we discussed the BV-BRC website, searching for data in the BV-BRC, how to make a workspace, data tables, and any conclusions from that. Then our second, we focused on the BV-BR website and the outbreak pages more specifically, and generating phylogenetic trees in the BV-BRC and the phylogenetics within those outbreak pages. So if you're new to the BV-BRC, we are here to provide a one-stop shop for data about human pathogenic and related bacteria and viruses into a scalable resource from comparative bioinformatics, large-scale data mining, multi-scale systems biology exploration, and machine learning to support the evolving needs of the infectious disease research community. And we're supported by NIAID to do that, and we are here to make the BV-BRC as useful as it can be for you, so please feel free to give us any feedback to make the BV-BRC even more useful for you. So some key features of the BV-BRC: we support diverse bacterial and viral communities, really any bacteria or virus that has a human pathogenic component. You'll find that data and tools to analyze it on the BV-BRC. We have a unified data model and real-time data integration processes. So if you're looking at one bacteria or one virus, you should see the same format across the website. So that model is continuous and should be easy to understand once you figure it out for whatever virus or bacteria you're interested in. We have efficient data management to support exponential growth of pathogen data. Thinking back to the beginning of the SARS-CoV-2 pandemic, we had no SARS-CoV-2 genomes, and now we have millions and are able to support that with ease. We consistently and accurately annotate genomes and other derived data types to make all of the data coming into the BV-BRC as useful as it can be. And another way that we do that is through automated and manual curation of metadata, and have modular, interoperable, and compute-intensive high-throughput data analysis services, and that's what we're going to talk about a little bit more today. Then we have integrative analysis of multi-omic system biology data using visual analytical tools, and phylogenetic and epidemiological analysis for rapid outbreak response. As a user of the BV-BRC, you have a private workspace that you can analyze, share, and publish data. You can keep it private to you within your lab group and beyond. And we also have programmatic and batch access that you can access BV-BRC data via APIs, the command line interface, and FTP. Finally, we do outreach and education like this webinar right now. So I wanted to highlight all of the BV-BRC services. We're going to go into some of these with more detail today, but by and large, we have services that span genomics, phylogenomics, protein tools, metagenomics, transcriptomics, utilities, some viral-specific tools, and then all of our outbreak tracking. Here are just some pictures so you can kind of begin to understand images that you can pull from the BV-BRC. So we have a multiple sequence alignment, our phylogenetic analysis colored based on some metadata feature, our surveillance data that links to a map to be able to view that, and then our protein structure viewer to view structures. So when we're thinking about how the BV-BRC responded to the H5N1 outbreak, we share relevant H5N1 news with the community, and create an infectious agent primer that you can see here in our newsletter. If you're not currently signed up for our newsletter, please do so you can keep up with everything that we're doing. We assisted in the submission of thousands of sequences for 2024 and generate phylogenetic trees that we continue to update, and those are on our Outbreak pages that we talked about last week. We created those H5N1 Outbreak pages, and we also released sequence feature variant type data for influenza, so you can view that on the BV-BRC. And I left a point for anything else you want to see from the BV-BRC. Please let us know so we can make it as useful as it can be for you. Thinking about the monkeypox virus outbreak, we had a very similar response. The only additional thing that we've done for monkeypox virus was add our subspecies classification tool and then classified all the monkeypox virus genomes in the BV-BRC to the clade level. So now that data is annotated and accessible for you to use. And we just, as of this morning, released the sequence feature variant type data for monkeypox virus. So I'll show you that in a little bit more detail. But the BV-BRC team is continually trying to meet the needs of the community for rapid outbreak response. So again, if there's anything else you want to see from us, let us know so we can further support you. So broadly, when we're thinking about tools for analysis, you can use any tool on the BV-BRC in the tools and services section, which here we're just to orient you away from the presentation. This is the homepage for the BV-BRC. And then on the top bar, we have our tools and services menu. And that was like that screenshot that I showed you a few slides ago. All of these tools and services are welcome for you to use. I'm just going to highlight a few to better understand the capabilities of those services and how a general BV-BRC tool and service works. So to start, I just mentioned that we had added the subspecies classification capabilities for monkeypox virus, and we also have that for influenza. And the subspecies classification tool uses pplacer and a curated reference tree to denote a subspecies classification. So you'll see on the right side of this slide an output of a subspecies classification job, which will have the target sequence where it falls on that reference tree, and then it will generate the designation for whatever virus that it is. This tool can be run on multiple sequences and is very efficient. And the one thing to note with our subspecies classification tool, as we go to our tools and services, it will be under viral tools, subspecies classification. There are different inputs depending on the virus that you're interested in. So if we're interested in influenza H5 classification, we recommend, or you should be using the hemagglutinin gene for that classification and genomic RNA. Then if you were doing a monkeypox virus, you can use the complete genome or a big piece of the genome to get that classification run. You can do one sequence or multiple. You can also just upload a FASTA file to your BV-BRC workspace. That's something I haven't shown on these webinars, so I'll take a second to show you how to upload data into your workspace. So there's a few different ways. The first one is clicking into your workspace. So we'll go to my webinars folder. And I'll make a new folder for this one. you can click that add folder and that will create a folder. And in this folder, you can upload any type of file that you want. You can upload a PDB file or a FASTA format, whatever. Really, any of these file types you're able to upload. And I know that this is an unaligned FASTA, so then I can do featured, and I know that it's DNA, so can and it will tell you that the typical file types. So if it was a GenBank file or a doc file and so on, that will let the BV-BRC know what kind of file it is, and then it will give it the right type. So then you can upload that, and then that will be ready for you to analyze. So that can then be used on any of our tools and services, depending on what type of file you need. But that's kind of our basis for the subspecies classification tool. Also, all of our tools and services have reference guides and tutorials that you can access here at the top, or by clicking the link that says quick reference guide or tutorial. This will bring you to our documentation pages. We'll tell you where the tool is, how to work it, what the output will be like, what files you'll get when you run that job. And that, again, is the same across our services. So continuing on with the presentation, we'll look at our Metacats tool, and this is a metadata-driven comparative analysis tool, and it looks for positions that significantly differ between user-defined groups of sequences. It's important to understand that there may be biases with the Metacats tool that may be due to covariation, codon biases, differences in genotype, geography, time of isolation. Those things can all affect the underlying statistical assumptions, but basically you'll have two groups, two or more groups, and then seeing if there are any statistically significant changes in position between those groups. So at position 225 in this example, there 28 from group 1 had a V, where only 22 in group 2 had a V, where 6 had an A. So this can be useful for finding potential PCR targets or other specific base pair changes or amino acid changes that will potentially impact different places in time, geography, years, things like that. So the Metacats tool also is under our protein tools. They're same quick reference guide and tutorial. You set the parameters at the p-value and then where you want your job to run. You can auto group based on some of these metadata features. You can have feature groups as an input or an alignment file. The auto grouping allows you to work on common BV-BRC fields. So this is another tool that might be useful for anyone during their analysis during an outbreak. In our first webinar, we talked about our first fall webinar, we talked about the sequence features, and these are a formal definition is that it's a specific string of amino acid positions that make up a defined region, a particular reference strain possessing some characteristic of interest. So this is an amino acid motif with the functional significance and conserved with respect to a reference strain ortholog. They're linear, contiguous sequence domain or discontinuous sequence structural domains, and they may overlap and have no size limit. And so our sequence features fall under 4 main categories: functional, structural, sequence alteration, and epitope. We talked about this in relation to influenza, but now we have sequence feature and variant types for monkeypox virus. So each sequence feature has variant types, and so the variant types explain the sequence variation for each sequence feature, and they're computed after multiple sequence alignments of all relevant virus genomes available in the database. And all virus genomes with amino acid sequence identical to the reference genome in that region are characterized as belonging to variant type 1. So when we look at the sequence feature data in a second, we can begin to see how anything like the reference will be variant type 1, and then subsequent variant types are ordered based on decreasing frequency of representation. So you can use the sequence feature variant type data for the identification of amino acid motifs in virus proteins that impact biological function and virus host interactions, tracking variations, investigating genotype, variant type grouping, phenotype, metadata characteristic associations. So our sequence feature data is accessible in 2 places. The first is under our searches, advanced searches tab. So we talked about influenza A a couple weeks ago, but now we can see the sequence feature variant type data for monkeypox virus. And so our last webinar, we talked about the OPG057 gene, the palmylated EEV membrane protein. So we can click on this specific gene protein. We'll keep all of the sequence feature types and the clades, and we'll search the database for that. So these are all sequence features that are associated with this gene. If we were interested in specific types, you can click on any specific row. You'll see the gene, protein name, sequence feature, and the identifiers, what reference it's with, the positions, where this information came from, and then any publication information. So if we're looking at this position 174, K174N, it's a potential antiviral resistance mutation, potentially leads to TPOXX resistance. This mutation has been observed and tested only in combination with other resistance mutations. So these comments are a great place to understand why this variant is on the list, but then we'll go just click through a couple others so you can see different interesting variants. And so this will be all of the monkeypox virus genomes. So these are not specific to 2024. You'll have to do a little digging to find sequences, specific sequences, but it is a great place to get an overview of sequence feature variant types. So if we click on this one, which is also important for TPOXX resistance, which makes sense for this gene, you can click on any of the sequence features and then click on variant types, and this will pull up a table of the actual variant types. So we'll do that. And for this specific sequence feature, we have 4 variant types. So most are like the reference. 15 have that N to, or 15 have an N to a D, and some have an unknown, but you can click on the ones that are any one of these to further analyze it. And that will pull up all of the sequences that have that specific variant type of interest, and then you can use those for further analysis. Rashni, is there anything else that I should add about the sequence feature variant types?
You can tell about the find option.
Oh, maybe you can tell.
No, it's go to the variant types, and if you are looking for a specific variant type, now for this you have only one position, but maybe like if there are more than one position, then you can look for a specific combination of amino acid through the search function.
Let's do a bit longer one. So you can begin to change those to look for the specific one that you're interested in, for example.
This is populated with the VT1 initially, and you can modify it based on your interest to look for any specific combinations.
I think we went a little too big for that sort of search versus too small, so maybe we can find. But anyways, that find feature exists. You can also look for keywords. So since these are all the same gene, but we do. So that's another way to find the keywords of interest. You can see that the spelling is important. So I pulled up one sequence feature. It was actually this one, which is also important for TPOXX resistance. So if we click on that variant type, this A288P might be something that is a target for someone's analysis. So thinking about using that as a way to further review our conversation from last week, but also connecting the sequence feature variant type data into other tools within the BV-BRC. We can then take a OPG 057 sequence and target this A288P with our primer design. So as you might know, this is a highly conserved gene. So I picked a nucleic acid sequence for this and thinking about designing primers to target variation at this position. So the nucleic acid side around 864. So I took the sequence of interest and put it into our primer design service. We can, I'll model how to do that, but so I'm going to just search for a sequence. And again, since it's pretty conserved, this broad search is going to be more than enough for our needs. So you can see we have this genes, nucleic acid. You can pull out the FASTA from this view button. So then I'll take that sequence, copy it. Then I will go to our primer design service, and we can upload FASTA files for an input for the primer design service, but we can also just paste the sequence. The identifier goes up here, and then the sequence text fits within there. You can manually mark a region by sticking in these symbols, but you can also target a region or exclude a region or include by putting in the variables as they're put here. So for excluded regions, if you include, the numbers 401,7, 68,3, that will forbid primers in the 7 bases starting at 401 and then 3 bases starting at 68. You can exclude 2 regions. As I mentioned for this example, we're going to target around 864. And so that will be the position that you're interested in, how many base pairs around it. So we'll do 864, 5. And that should target that specific region. You can add some advanced options for primer temperature, GC content, and so on. I'm not going to run that job again, but I ran this job here. And you'll see the standard output for a primer job. You can see that these jobs run very quickly. It's done in 3 seconds. I'll show you the input text output, FASTA file with the primers, and an HTML table. And I think I did 3 instead of 5 this time, but you can see where the forward primers, the reverse, the internal oligo, start and length of the primers, melting point, GC contact, and it gives you a few different options for those primers. You can download this table or the input and output in FASTA file. So that's a quick overview on our primer design service. Then we also have some services like Fastq Utilities, viral genome annotation, multiple sequence alignment, and SNP variation analysis. So you can use Fastq Utilities for aligning, measuring base call quality reads, and trimming Fastq read files. Viral genome annotation uses Vigor4 for the annotations, and we talked about the multiple sequence alignment a bit last week, but again, annotation service is under genomics and takes a config as a input file. If we're thinking about these viruses, you would use viruses as the annotation recipe. Can do monkeypox, viruses, the taxonomy name, and identify it with something and run that job. Our Fastq utilities can be done in a pipeline form, so you can trim first, then run a FastQC, then scrub out any human sequences. Maybe you want to do that first, then trim. Really change these around based on whatever your need is. Our multiple sequence alignment and SNP analysis is under protein tools. And that takes an input of a feature group, a viral genome group, a DNA or protein FASTA file, or just an input FASTA sequence. And you can align it to a reference. That's either the first sequence or input a reference sequence. or none at all. We have a few influenza-specific tools and services that I'd like to highlight. So we have an HA subtype numbering conversion that takes influenza HA protein sequences and converts existing HA position number to a different HA numbering scheme using David Burke and Derek Smith's method that uses both sequence and structure information to propose positions of functional equivalence across the different HA subtypes. So you can input protein sequences, then the sequences are blasted against the Burke reference sequences that will determine the best reference subtype using the HA numbering pipeline. Then we'll generate a pairwise multiple sequence alignment using the reference protein sequence. And the alignment will generate a mapping between the input sequences and the blast reference sequences and gives you the selected HA subtype positions. So that tool is under our viral tools and takes in the input sequence and whatever numbering scheme that it would like to be converted to. And the paper for this proposed scheme is here. And again, all tools have the quick reference guide and tutorials. The next tool I want to cover is our sequence submission tool, and this is to validate and submit virus sequences to NCBI GenBank. There's two main steps in the sequence submission process. The sequences are validated, so compared against GenBank submission standards to identify any sequencing errors. And unlike other BV-BRC services, this one has some validation with us to make sure that the sequences are ready to be submitted. If there are any issues, then we'll come back to you with those issues so that there will not be any issues when they're actually submitted to GenBank. And then sequences are annotated using VADR Flan for internal use by users. So you specify sequence submission parameters, include all of the submitter information, and have a metadata file and a FASTA file, and you annotate and validate those sequences. There will be 13 output files, including a sequence validation report. This CSV file shows the validation result for all sequences, and if there are any that have any issues, then you can go back and check those. And then once you are happy with all of your sequences, you can notify our team with the submit sequence button, and then that will get them ready for GenBank submission, and we will submit them. So this is also under these viral tools. We'll auto-populate your submitter information based on your BV-BRC account, and you can add whatever information that you want. You can enter just the sequences or them as a FASTA file, and you have to include this metadata file. And the template for the metadata file can be found here. So click here to download the example files. So we have a standard detail Excel file, example FASTA file, and metadata template. So if you download this file, you can see the standard details with some explanation. So hopefully this is as easy and useful as it can be, but again, feel free to ask any questions if you have any with your submission. Just a reminder, this folder is going to be, can get directed to it from click here to download the templates. Okay. to our presentation. The last tool and service component that I want to talk to you all about is thinking about larger jobs. So BV-BRC supports bulk analysis, which might become a little cumbersome with our web interface. So if you're a little bit more computationally savvy and interested in doing large-scale analysis, recommend using a command line interface or API or our FTP. And I took this screenshot from the website that included this batch access, but also the tutorials in one. So each of these bulk analysis has really strong documentation. So going from the home page to command line interface brings you right to the documentation for that, tutorials for each installation to actually using the tools. So this should also be pretty user-friendly, and some commands exist. And going back to the home page in either one of our Outbreak tabs, you can also see some commands here to download annotations, search genomes, download genome data, and submitting a genome annotation job. If there are any commands that you feel like you need help with or missing, please let us know so we can help you get that. And there's a few ways to ask us for help. So you can, under our About section, click Contact Us, and then that will submit us ticket. You can also email us at help@bvrc.org. Be happy to answer any questions there. And then if you have a job that you expect to work, but it doesn't, the failed job, you can report an issue. And then that will give all of the parameters of that job so you can better understand what went wrong and help you get an answer. You can also report an issue with a job that worked. If you got an output that you weren't expecting, can provide a question or relevant feedback, and that will submit a ticket to help us help you. So I want to close by mentioning we covered the outbreaks briefly, some tools for analysis in the BV-BRC, monkeypox virus sequence feature data, some influenza-specific tools, and a very quick overview on our bulk analysis. So next, I just want to offer some tools that you can look at things further. We had a Back to Basics webinar series that covers searching for data, annotating viral genomes, building trees, sequence submission, and analyzing protein data, and more specifics. All tools have help documents. There's Archaeopteryx-specific help documents. Please follow us on LinkedIn. We just talked about the command line help documents, and the BV-BRC team is very grateful for your use, and we want to continue to bring you tools and services as you need them.